Hello, good afternoon, everyone. I hope you had a good break last week and you are all ready for the second half of the course. So, second half is pretty much similar to the first half in terms of the structure. We will have five lessons, 6 to 10, and there will be two 10% quizzes, one 10% lab, as well as a final test, which is going to be um, the 20% test, right? And as I mentioned to you previously, the second half is standalone compared to the first half. So all the stuff that I already test you on will not show up again on the final test. Okay, so hopefully that's a little bit of a relief for at least some of you. So let's get started. Lecture 6 is about cellular reproduction. We are going to be taking quite a bit of notes today, so make sure you have your colors and a lot of papers with you. So this lecture is separated into two halves. The first half is about how DNA replicates. And the second half, which we'll do on Thursday, is going to be about how a cell actually divides, okay? And what happens if the division process contains errors? It could lead to things like cancer and so on and so forth. So that's going to be on Thursday. We'll begin by looking at some brief history regarding DNA, how it was determined to be the molecule for inheritance. We'll look at how people figure out um, how DNA copy themselves. So there are a few key experiments that we're going to go through. So that's going to take us like the first hour. Okay. In this picture, we can see there are two interesting things here. These little green uh, structures here, these are actually a type of virus. Okay, and the virus is landing on a bacterium right here. So this is a bacteria. Bacterium is singular for bacteria. And the virus is injecting something into the bacterium. And that something is going to hijack the bacterium's machinery. So it's going to take over its ribosome and things like that. And it's going to start forcing it to make more viral particles. Okay, and then the viral particles come together and assembles into a full virus, which then gets released from the bacterium. So people in the past, they understood this concept, but they were not sure what was injected into the bacteria. Okay, whatever that thing is that gets injected into the bacterium, that is the molecule that is responsible for heredity. Okay, that's the stuff that allows the virus to make more virus. So if they figure out what that is, then they could determine the molecule for heredity. I mean, like we know now that it's DNA, but back then in the 1950s, there were two camps of scientists believing different things. On one side, scientists think protein is the molecule of heredity. Okay, on the other hand, some think DNA is the molecule for heredity. Because as you know, chromosomes is a combination of DNA and proteins, right? So they're kind of intertwined together, and it's not always easy to figure out which of it's responsible for passing on the information to the next generation. Okay, so that's what I mean when I say hereditary material, right? It's the information, the genetic information that gets passed on to the next generation, whether it's a cell dividing or whether we're talking about like an entire organism, like when we have kids, what we pass on to our kids, right? Like, is that proteins or is that DNA? That's what they're trying to figure out, right? And so the first experiment that we're going to look at addressed that question, and it was done by two scientists shown here, Hershey and Chase. Okay, so here we go. first note of the day. Hershey and Chase experiment. So the purpose of Hershey and Chase experiment, purpose is to determine whether proteins or DNA is responsible for heredity. Okay, that's the purpose. For their experiment, they used a virus called bacteriophage. Okay, which is those green things that I showed you in the PowerPoint slides. Okay. So the bacteriophage has two parts to it. Okay. There is a protein coat and inside the protein coat there are some DNA. It's a DNA core. So that's basically the bacteriophage. And the bacteriophage, they don't infect humans, they only infect bacteria. So it's relatively safe to handle. So let's talk about what they knew. Okay. Hershey and Chase, what did, or like at the time in the scientific community, what do people already know? Well, they understood that something is injected into the bacteria by the viruses. Okay. That something is responsible for making more viruses. Right? So that something is responsible for making more viruses. That is the molecule responsible for heredity. Okay, so this is basically like the premise that they were operating on. Okay, so they understood this thing. They just got to figure out what went inside the bacteria. Okay, I'll give you a moment to catch up. All right. So, Hershey and Chase, they created They created 2 batches of bacteriophage. Right? Batch number 1. These were grown in sulfur 35. Now, from your chemistry class, do you remember what the normal mass for sulfur is? Like if you look it up on the periodic table, anybody at all? No, that's okay. All right, so sulfur is normally 32. Okay, so this one is sulfur 35, which is heavier isotope of sulfur, and it turns out to be a radioactive isotope. Okay. And the whole point of growing them in radioactive sulfur is for you to be able to keep track of where things go. Okay. If they're just ordinary atoms, then it's hard to know what goes where. But if they're radioactive, then you can detect the radioactivity and keep track of things. So the virus has a protein shell, protein coat, and a DNA core. If you grow them in radioactive sulfur, is the DNA going to become radioactive or is the protein going to become radioactive? What do you think? We're going to grow them in radioactive sulfur. Is the radioactive sulfur going to be incorporated into the virus protein or is it going to be incorporated into the virus DNA? Anybody can tell me that? Okay, so Peace said protein radioactive, and Micah said the same thing. That's correct because protein contains traces of sulfur, right? But DNA does not. Right? So let's draw that out. We're going to have the radioactive protein shell, protein coat, and then we're going to have the regular DNA inside. So protein coat is radioactive. Now, they needed another way to track the DNA. So batch number 2, what is a good element to tag the DNA with? What's something that makes up DNA but not protein?
Phosphorus?
Yes, that's exactly it, right? Very good. So these are grown in phosphorus 32. Okay. Normal phosphorus is 31. So these are also radioactive isotope. Okay. So in this case, the protein shell of the virus will be ordinary, and then you really need color for these notes, right? For it to make sense. And then you will have the radioactive DNA. DNA core is radioactive. Okay, so there you have it. 2 batches of um, viruses. All right, the experiment. Okay, so what did they actually do? Well, it's pretty simple. They infected bacteria with batch 1 or batch 2, separated the virus viruses, I guess, and bacteria using centrifugation. Okay. So centrifugation basically means you spin it at really, really high speed. And when you do that, you basically separate the contents by mass. Okay, so separate viruses and bacteria using centrifugation, which cause the heavier components to sink to the bottom. Okay, so I can draw a picture for you. Okay, this is a test tube, and the solution they put in would have the virus and the bacteria, and then they could put it in a machine that spin it at really, really high speed, and then it would separate them into two portions. Like I said, the heavier stuff sinks to the bottom, and then the lighter stuff, it's gonna go to the top. The best way for you to visualize this is think of mixing sand and water in a bottle. Okay, it's gonna look very, very murky. You let it sit there for a bit, the sand eventually will all sink to the bottom, right? Because the sand is heavier than the water. Um, and you can speed up that process by spinning it at really high speed. That's what centrifuge means, okay? Centrifugation. Yeah, exactly, right? That's how they separate blood. We actually will learn a lot more about that in the second semester, but thank you for bringing that up. So I want to ask everybody, is the bacteria heavier or is the virus heavier? Which one is heavier? Virus or bacteria? Which one is bigger? Is the virus bigger or is that, I mean, look at the picture, right? Look at the picture here. These are the viruses. This is bacteria. Right? Bacteria is like much bigger compared to viruses, right? I mean, not everything that's bigger is heavier, but in this case, bacteria is both bigger and heavier. Okay, so that's gonna sink to the bottom. This bottom part here, this will be the bacteria plus whatever was injected into them. Okay. So remember, they knew something got in the bacteria from the virus. They just don't know what it is. But whatever got in, it's going to stay in the bacteria. And when you spin it, it's going to sink to the bottom with it. And over here, the top part, that's the portion of the virus that stayed outside. They did not go into the bacteria. Does anybody have any question about this setup and the meaning of the test tube before we continue? Okay, so that was the experiment. Let's take a look at the result. Results. So let's check the batch one. Batch number one. These are the viruses that have the radioactive protein. Okay, radioactive protein, and they infect the bacteria with them, and then separate them, spin it down, and detect the radioactivity only at the top portion. Radioactivity for the top portion. What does this mean? What does it mean? This means the protein of the virus did not enter the bacteria. We can say viral proteins did not enter the bacteria because it stayed outside. Top is part of the virus that didn't go in. What this does not show, does not confirm, is that the DNA went in. And we actually have no idea where the DNA is based on this result. We are only tracking the protein, right? Maybe the DNA didn't go in either. Okay. That's why they needed the results from batch number 2. Okay. Remember, this is sulfur 35. Okay. And then the batch number 2 is phosphorus 32. Okay. So this time, the DNA are being labeled. Okay, and let the virus infect the bacteria, spin it down, and they detect the radioactivity only at the bottom. Or the bottom portion. Okay, so what does this mean? This means the viral DNA entered the bacteria. Okay? Again, this second result alone is not enough. It only tells you the DNA went in, but it doesn't tell you where the proteins are. Maybe the protein went in as well. Okay, that's why you need to have the results from both batches to show that no, the protein actually stayed outside and only the DNA went in. So when taken together, the conclusion of their experiment is that DNA and not protein is the molecule for heredity. And that is the experiment from Hershey and Chase. Any questions? Okay. I'm not going to ask you to explain this whole thing, but you should understand the mechanism behind it so you can answer some of the questions on the quiz. All right. This is just some notes about the Hershey and Chase experiment with the bacteriophage here. You can read it yourself. And this gives you a visual representation of what actually was happening. We have the sulfur 35, which is just labeling the protein code. And then we got the DNA injecting into the bacterium. Afterwards, when you separate it, right, the virus goes to the top, and you will only have the radioactivity at the top because the proteins are being labeled, right? But when you have the second batch where you label them with the P32, the DNA gets injected into the bacterium, and when you try to separate it, you will have the radioactivity at the bottom. So once again, when taken together, these results confirm that it's DNA that's responsible for heredity instead of proteins. Okay, so now people know that. The next thing they wanted to do is to determine the structure of DNA, and there were a few people who contributed to this. Rosalind Franklin, she used X-ray to study the structure of DNA, and this is like one of the famous pictures from her data. Okay, when she looked at the data, she realized DNA is actually a double helix. Okay, so that's a single helix, and then you have another helix here. So that's a double helix. That's DNA right there. Okay. Unfortunately, she died of cancer before she can finish her work. And later, what happened was Watson and Craig, which are these people here, they took her data and built upon it, and they were able to create the first scale 3D model of DNA. They later won the Nobel Prize for that. But people are saying, it should have been Rosalind Franklin who won the Nobel Prize because it actually was her data that allowed the discovery of the structure of DNA. Okay, so what do we know so far? We know DNA is hereditary material. We know the DNA is double helix. Um, and then the next thing people try to figure out is the model of DNA replication. Okay. So at the time, there were three proposed models of how DNA copy themselves. And later on, these people, Messelson and Stahl, they are the ones who did an experiment that would confirm which model is the correct one. But before that, the purpose of DNA replication. When we were learning about cells last first half of the course, part of the cell theory states that all cell comes from the division of pre-existing cells, right? So in order to get more cells, you must take a cell and then it needs to divide and it creates more cell. So here we have a cell and we call it the parent cell. Don't be confused, okay? Like when we say parent cell, we don't mean the cells of your parent necessarily, okay? It could be a cell in your body that's ready to split, ready to divide. That original cell, we call it the parent cell. And for simplicity, there are two strands, two chromosomes here, okay? Two DNA, the blue one and then the purple one. And so before the cell divides, it must undergo DNA replication to copy all of its chromosomes. So you have two blue and two pink. And then when it actually divides, which is called mitosis, that's the second half of this lecture, you will have two daughter cells. And these daughter cells are going to be genetically identical to each other because you've copied all of the DNA prior. Okay? So that's why we need to do DNA replication. If we don't copy the DNA every time the cell divides, you're going to lose half of the genetic material, and that is not functional, right? All right. So there were three proposed models of DNA replication: the semi-conservative model, conservative, and dispersive. In the semi-conservative model, here is the parental DNA, the original DNA shown in pink. And what the semi-conservative model says is you will separate the two strands and then you will use each strand to build a missing half. Okay, so the blue is representing new materials. So semi-conservative, each copy would be made up of 50% old material and 50% new material. 50% original, 50% new, and it's going to be restricted to half strand. Essentially, one strand is old, one strand is new. That's semi-conservative. Conservative model say that this is the original material, and when you copy, get to keep the entire original double helix, and then you're gonna make a complete new one. And in dispersive model, it's kind of like, pick and choose. You get some from each strand, and then you build the missing half. Right? To help you understand, I've created this picture here. Right? This took me like half an hour to make, actually. Hopefully, it's going to be helpful. There is the original copy of the picture. And in this case, it's like putting it in a photocopier. Right? You make a duplicate of the original, and then it looks like this. So do you think this represent conservative, semi-conservative, or dispersive? What do you think, huh? Let's do a poll and see what you guys think. The first picture here, is it conservative, is it A, semi-conservative, B, conservative, or C, dispersive? This one. Right? Semi-conservative, conservative, or dispersive? Okay, thank you. Not a lot of answers here, but this would be conservative. Conservative. Okay, because you are preserving the entire original and then you're creating a duplicate. Okay, next one here, it's like we cut the original in half and then we use the original to create the missing half. Okay, because it's mirror image, so you can create the missing half. And same thing here, we use the original and then you create the missing half. This one is semi-conservative. And the last part here is like you take bits and pieces from here and there, and then you patch it together, and that would be dispersive. Okay. Both of these models, the semi-conservative and dispersive, they are each copy is like half new, half old. But which half is new, which half is old, that's what distinguishes them. For semi-conservative, the entire side is original material. Whereas over here, the original material is kind of all over the place. It's like a patchwork of old and new mixed together. Okay. If this is still confusing for you, let's write it out. Maybe it will be a little bit more clear once we draw it out. Okay. So let's write down three models of DNA replication. Okay, you need two colors for this to make sense. Okay. I'm just gonna make up some DNA strand. A, T, C, G, T, A. So the other side must be T, A, G, C, A, T because of complementary property of DNA, right? So I'm just gonna make three of these things. Okay. So let's see what happens if it is conservative. Okay, maybe don't copy now. Just watch, and then once you understand, you can copy it later, from the notes or something, right? So take a look here. Conservative, what does it mean? Conservative means this. You take the whole thing, and then you just make a copy of it. And then the new stuff, I use blue to represent it. That's conservative. Understand? You literally just copy the whole thing. So you would have original material in one DNA molecule and then new material in another. Okay, so that's conservative. Let's take a look at semi-conservative. Okay, watch. Okay, don't copy. So in this case, what happens is you will separate the two strands. You separate the two strands like that. And you will use the complementary rule to build a missing half. So your body knows A pairs with T, T pairs with A, C pairs with G, G pairs with C, and so on and so forth. Same thing over here. T pairs with A pairs with T, so on and so forth. So in the end, you get two copies. These are the same as the original, okay? But each copy will have one strand of original material and one strand of new material. That's semi-conservative. Any questions before I do the last example? If this doesn't make sense, you gotta ask because it's only gonna get more complicated.
I do have a question.
Sure.
Because it says like it's the three proposed models of DNA
Mhm.
replication, does that mean like only one of them is actually correct?
That's right. So after we explain this, we're gonna look at the Mesosen install experiment, and then the results will tell you which one is correct. That's a good question.
Okay, thank you.
Yeah. Yes, Amr, go ahead.
How is the new material obtained?
That is a good question. Yeah. Um, so the new material is the nucleotides, right? Okay, so there will be nucleotides floating around in the cell, right? In the body, for example, when we eat the food, right? Um, it gets broken down into building blocks. We learned about this last time, and some of these building blocks might be amino acids that we can use to build proteins. And some of these building blocks are going to be nucleotides, which we can use to build new DNA. Does that answer your question?
Yes, thank you.
Okay, so here the last model is dispersive. Okay, so dispersive, it's like this. All right? It's you're gonna take a bit from each. It's actually really hard to do on the thing here, but just let me try. Okay. And then we're gonna take maybe these three things, and then I'm gonna use it over here, and we're gonna take these three things, and I'm gonna put it over there, and then the T is gonna go over there. Essentially, what the dispersive model says is, okay, we just randomly take nucleotides from this side and that side, and then afterwards, we're just going to build whatever's missing. So, A here, I know it goes with T. T goes with A. G goes with C. All right. So in the end, you're going to have the strand that's mix and match of new and old material. Okay? I mean, the semi-conservative is also mix and match, but it's restricted to one side, right? You get one entire strand of original and one entire strand of new stuff. But here, the new and the old are kind of like, uh, zigzag, if you would, throughout the entire DNA. So those are the three proposed model. Okay, and as mentioned earlier, the people didn't know which one is correct. These are just ideas of how DNA um, were replicated. Okay. And so the experiment that would answer that question, that would figure out which is the correct DNA model, would be the Mendelssohn and Stahl experiment. Mendelssohn and Stahl. There we go. Ideally, you would have three colors: blue, red, and purple would be great. If you don't have purple, I guess something else is fine as well. So this is Mendelssohn and Stahl experiment. Purpose. Okay. The purpose of the experiment is to determine the correct model of DNA replication. Right? Was it conservative? Was it semi-conservative? Or was it dispersive? Right? So they got to figure it out. All right. So how did they do it? They grew E. coli in heavy nitrogen. Okay, please use colors like I do so that it makes sense. For several generations. Okay, so regular nitrogen is 14. Again, this is something you can look up on the periodic table. Have you guys actually learned that yet? I feel like no one knows what I'm talking about these past couple of days when I was talking about, regular nitrogen and 14 and stuff like that. Yeah, you have? Okay, great. Maybe you guys are just quiet. Okay. So regular nitrogen is 14. This is heavy nitrogen, N15. And this is not radioactive. Okay. Um, you can actually see DNA when you extract it in a test tube. Okay. It looks like a little bit of like shampoo flowing in the in the test tube. Okay, so anyway, they grew the E. coli. E. coli is the bacteria, right? Maybe you heard on the news last week in the US. There were like 50 people who got sick from E. coli because the burgers at McDonald's were contaminated. I think it was the onions actually, and they got really sick, right? So E. coli, right? That's the bacteria. So they grew E. coli in heavy nitrogen for several generations. This several generations period, I guess. This caused all of the E. coli DNA to be heavy. Right? So when the bacteria divide in a dish, you have to supply the nutrients for them to grow, right? So if you're only giving them heavy nitrogen, then they are only incorporating heavy nitrogen into the DNA that they're creating. So after several generations, all the DNA will be heavy, right? As you know, the DNA contains nitrogenous base. All these ACTGs have a lot of nitrogens in it. And so if you incorporate N15 into them, DNA is going to be heavy. Okay, what did they do next? Okay. They, as in Meselson and Stahl, transferred the E. coli back into regular nitrogen, which is N14, and allowed them to divide once. The E. coli DNA were extracted and separated by mass using, once again, centrifugation. Okay, you spin these DNA down and it will separate by weight. Heavy stuff goes to the bottom, light stuff goes to the top, and then if it's intermediate mass, it goes to the bottom. Okay, so that's what they did. All right. Now I'm going to do a side note here for you. Side note. There is the test tube. Okay, this test tube is not like empty test tube. Okay, it's like filled with like it's like a gel-ish stuff. Like it's like maple syrup. Okay, in there. Okay, so if you put things in, it's not just gonna fall right to the bottom. And then you can spin this thing, and like I said, it will separate them by mass. Okay, so this is the very important thing to understand. Okay, stay with me, and please ask me if you don't understand. If I have the original DNA from the E. coli that has been growing in N15 for several generations, if I extract those DNA and I spin it down on this test tube, do you expect the heavy DNA to be at the top, middle, or bottom? It's a rhetorical question almost. Where is it going to be? Yeah, it's going to be at the bottom, right? So you will be able to detect DNA down here. You can actually see it. Obviously, it's not pink, but I'm using pink to help you visualize it. So these are the N15 DNA. Okay, actually, you know what? I'm going to put it on the side. N15. What that means is both strands are made up of heavy nitrogen. Okay? What if I have regular DNA strand that's made up of just the regular nitrogen, N14. Well, I hope it's not too difficult to understand that it will be at the top because 14 plus 14 is 28, 15 plus 15 is 30, which means this is lighter, right? It's going to be at the top. And in the middle is where you will have a mix of N14 and N15. And you will have half N15 and half N14. It's not as heavy as 1515 and it's not as light as 1414. So it's going to be somewhere in the middle. Okay, so the DNA could look like this. Or, or it could look like this. It's actually hard for me to draw this next one, but it's like the dispersive stuff. Okay, it's a bit of, red and blue mixed together. Okay, does everybody understand the result here? I mean, these are not the result. This is just like the interpretation of like where the DNA is gonna go. You have to understand this before I show you the results. Any questions? No? Everybody's happy? I know it's a lot of information. My first day back from the break. All right, so let's take a look at what happens. Okay, I'm gonna have to go back to the PowerPoint slides. Just give me a moment. I am gonna have to copy some of this stuff over. You obviously don't have to draw this next part, but I assume you have the notes with you, so you can always refer back to it. Uh, just give me a moment. This does take some time. There we go. Okay, so this slide is from the PowerPoint slide, and basically this parental DNA, that's the N15, N15, right? These represent the DNA from the E. coli that has been grown in N15 for several generations. Okay, and then these DNA is the expected result after one division. Okay, so in the semi-conservative model, you separate the two strands, and then you're gonna build the new strands from it. So in other words, half of the strand will be N15 from the original, and since Meselson and Stoll transfer them to N14 and then let them grow for one generation, any new materials that they build in the DNA will be N14. So if you extract the DNA here and you spin it in a column, where do you expect the DNA to be at? At the top, at the middle, or at the bottom? I'm gonna give you a poll to see what the class thinks, okay? Please participate so I know. Is it A at the top, B at the middle, or C at the bottom? Okay, we're talking about these strands right here. Is it top, middle, or bottom? Anyone else? Choose something. I will give you the reference here, the side note that you took. And that should help you figure out whether it's top, middle, or bottom. All right. So most people chose middle, and that would be correct. And if you did not choose middle, I hope you understand why it is middle, because half of it is 15, half of it is 14, right? So it's not as heavy as 1515, so it won't be at the bottom, but it's not as light as 1414, so it won't be at the top. It's going to be in the middle. Okay. What about if it's conservative model? Conservative model means you keep the entire original thing and then you make a copy of it. And if you extract the DNA and spin it down, where do you expect the DNA to be. Okay, it could be at more than one places. Okay, so please tell me, is it top, middle, or bottom? Okay, choose all the correct answers. Okay, so this is heavy, which means it's got to be at the bottom, right? And then this here, this is light, so it's going to be at the top. Okay, it's not in the middle because the mix is not within the same DNA molecule. Right? It's only going to be in the middle if the same DNA molecule has a combination of 15 and 14. All right? So that's that. And the last one here. What do you expect to see if it's dispersive? Where is the band? Top, middle, or bottom? Yeah, so it's a combination of 14 and 15, right? Which means, of course, it's going to be in the middle. Right? So these are the expected results if the model is semi-conservative, conservative, or dispersive. All right, so let's see. Result after one division. Okay, these are the expected result. The actual result was that they only saw one band in the middle. So what does it mean? Okay. Only saw a band, a DNA band in the middle. So what does it mean? Okay, Cheyenne said that means it's semi-conservative. Anyone else? What does it mean? Okay, pizza is dispersive. Anyone else? Oh, or dispersive. Okay, yeah. Yeah, you're right, okay? It's either semi-conservative or dispersive. Right? We don't know. So only saw a band in DNA in the middle. Okay, so conclusion, therefore, conservative. Conservative model is incorrect, but cannot tell if it is semi-conservative or dispersive, right? They don't know. So next, they let the E. coli to grow, to divide a second time before extracting the DNA. Give me a second, just copy the stuff over. Yeah. So let's go through the expected result. This is semi-conservative. This is conservative, which we already know is wrong, and then this is dispersive. Okay. So let me go back to here to show you. Okay, so if you focus on this DNA, if it's semi-conservative, means we're gonna separate the red and the blue. Okay, separate the red and the blue, and then from the blue you will build the missing half using more blue. That's why you have that. And then the red, you will build the missing half using blue, because only blue is available, right? Blue being the N14. Okay. So that's the outcome of semi-conservative model. If you extract the DNA and you separate it, what's the expected result after second division? Well, think about it. You will have the N14 at the top, right? N14 at the top from these guys. And then you will have a middle band from that and that. For conservative, even though we already know it's wrong, but if it was in the second division, you would have the 14 at the top and the 15 at the bottom. And what about dispersive? Where would you find the DNA in the dispersive model? Middle, that's right. Just the middle. Okay. So result. Actual result was that they saw a band at the top, and the middle. All right. So confirmed. The correct model for DNA replication is semi-conservative. Okay. All right, I'll zoom out a little bit in case you need to copy some of the stuff. Let's take a break. We'll resume at 1:35. Okay, see you guys in a bit. Hello, we are back. So there is question, private message say if I could explain how DNA replication for the different model for the second time. Okay, so like take a look at I guess we'll just do semi-conservative because that's what the correct model is. Okay, so this is after one round of division, right? You separate the two strands and then you build the missing half with the blue material. And now if I want to go for another round of division, look at this, okay? We're gonna separate this again, separate it, and then we're going to build a missing half. But all the new materials are blue, right? They are nitrogen 14. And so, you know, TAGCAT, and then over here, ATCGTA, right? That's why it's going to be half pink, half blue, and then blue, blue. And if you look at the semi-conservative model over here, you see, half pink, half blue, and then blue and blue. Does that make sense? Does that answer your question? If not, feel free to follow up. Okay. Okay. Wonderful. So those are all the experiments, all right, that we're gonna do today. We are gonna go back to the lovely PowerPoint slides here. Let's do some quick poll questions. In Hershey and Chase experiment, why was radioactive sulfur used? Let me relaunch the poll. Just look at the notes. We wrote it down. Okay, so most people who answer got it right. Is to label the protein coding of the virus, right? Because sulfur is found in protein but not DNA. Okay, over here, if DNA replication follows the conservative model, which we know it's not true, it's semi-conservative, right? But the question is saying if its conservative model is correct, then what would be the expected result from the first and second generation? Okay, so all you have to do in this question is to look at the notes that we took again, and we can see for, oops, this is a little bit frozen. For the conservative model, after one division, we would expect a band at the top and then the bottom. And then we follow a second division, we will continue to expect a band at the top and the bottom. So the correct answer for this question would be A, right? Top and bottom for the first, top and bottom for the second division. All right. So please familiarize yourself with these diagrams. I think the best way for it to make sense to you is for you to draw it out from scratch on your own, okay, with different colors and to see, separate them and what goes where, what color goes where. And after you do it one time, then it will make a lot more sense for you. Okay? If not, then please email me and we can set a time to talk more about it. All right. So now we are going to move on and talk about the actual DNA replication, the mechanism of DNA replication. Mechanism is just like a big word for the method, right, by which it happens. And much of the understanding of how this happens came from the study of DNA replication in prokaryotes. Okay, prokaryotes are like bacteria, right? And we are eukaryotes. So the process for us is a little bit different for a few things, but for the most part is quite similar. So we will learn the process in prokaryotes first, and then I will tell you the difference between what happens to them and what happens in us. Okay? So in this picture, what you're looking at is you're actually looking at DNA. Okay? Oops, this here, that is actual DNA. You cannot see the double helix. You cannot see the twisted up thing, but it is DNA. And here is a bubble, which is called the origin of replication. All right. So we're gonna talk about that in just a bit. All right. So in DNA replication, the parent molecule, which is like the original DNA, basically, it unwinds and the two daughter strands are built based on base pairing rule. Now, this part is a little bit tricky to fully grasp. So allow me to give you an analogy, I guess. Now, don't write this down. Right? So there is a strand of nucleotide here, ATCGCTA. If I ask you to copy this, if I ask you to copy it, what are you going to write in your notebook? Well, you're just going to copy it exactly as it is, right? You're going to write A, T, C, G, C, T, A. That's what it means when we say copy, right? But when your cells try to do it, copying means something else. They cannot copy word for word like that. They cannot copy letter to letter. If they see an A, it's impossible for them to write to copy as A, okay? When they see T, they cannot copy it as T. Instead, they have to build based on the base pairing rule. So when your cells see an A in the DNA, it would be building a T. When it sees a T, it will be building an A. When it sees a C, it will build a G and so on and so forth. Okay? So when it comes to DNA replication, you're not building it letter to letter. Instead, you're using the base pairing rule to build the complementary strand. Does that make sense? The copying of DNA is very accurate and very fast. Take E. coli, for example. There are over 4 million base pair in all of its DNA, and it's able to copy all of these DNA at a rate of 1,000 base pair per second. Yeah, that's very fast. That's like copying 1,000 of these ACTGs every second, right? For us, it's a little bit slower. We have about 3 billion base pairs in all of our DNA. And when we copy our DNA, it takes about, we are able to do it at about 40 base pair per second. Yeah? So, not as fast as the bacteria, but still quite a remarkable speed. Like, I mean, if you think about your typing, right? How fast can you type? Right? People can type like 40 words per second, right? 60 words per second, right? This is talking about 40 base pair per second. Right? Sorry, was it per second? Per minute, right? I think the typing rate is I think I got my the typing rate, I think I got it wrong. Okay. But then for the copying, it's 40 base per second. That's pretty fast. Okay. It's like typing 4 keys per second. It's like not possible, right? For us. All right. The process of DNA replication involves a lot of enzymes and proteins to make it possible. And here's a list of them. We will be learning about helicase, gyrase, single-strand binding protein, and maybe DNA polymerase 3 today. That's all the time we have. And then we'll learn the rest on Thursday. Okay. So now is time for another note. Okay, this one is a big note. So maybe take out a new piece of paper. I'll show you what we have to draw, okay? And then you can plan accordingly. It's just like from yesterday, maybe. Okay. So we're gonna be drawing this thing, okay? So make sure you plan some space for that. All right. Mechanism for DNA replication in prokaryotes. Okay, bacteria. Because it's easier to study them instead of studying animal cells. And we'll see that there are like striking similarities between what happens with the prokaryotes and with us. So let's start right there. Let's draw a bacterium here. And its DNA is actually circular. Okay, circular DNA. So if you zoom in over here, it's actually like the double helix, but it forms a loop that loops back on itself. It's like, I mean, you don't have to draw this. I'm just showing you. And this is like the base pair, right? The ACTGs. There you go. So that's like a circular DNA. Our DNA is not like that. Our DNA, eukaryotes, eukaryotic DNA. Ours is linear. Okay, it doesn't loop back on itself. And that actually creates a bit of a problem for us when we try to copy the DNA, okay? There we go. Now, in addition to the circular DNA, they also have additional small round DNA, rings of DNA. These are called plasmid. Okay, and plasmid gives bacteria special properties. Okay, like maybe it allows them to build an extra shell around themselves so they don't dry up as easily. This is true, like I'm not making it up, right? Maybe it gives them the ability to break down antibiotics. Okay, so we'll learn more about plasmid later on in lecture 9 when we do biotechnology. Okay, so let's take a look at the overview of what's happening here. Uh, Here are the original DNA. Okay, original DNA. I'm not going to draw the ACTGs. You know it's there, but this is one strand of DNA, the other strand, and they are bind to each other through the hydrogen bonds. When I taught you DNA last first half of the course, okay, there are two adjectives for DNA. Okay, the first one is they are complementary. The two strands are complementary, right? If you know the ACTGs, you can predict the ACTGs on the other side. What's the other word that I use to describe it? That they go in opposite direction. What's the term there? Does anybody remember?
Anti-parallel switching.
That's right, you got it. It's anti-parallel, right? It's like the highways, the roads are parallel to each other, but the traffic is opposite, northbound, southbound, right? Except we don't use north and south to describe the DNA. Instead, we use these numbers, 5 prime to 3 prime, and then on the other one is 3 prime to 5 prime. Don't worry about why it is 5 or 3. It's like a chemistry thing. Just know that if one strand is 5 to 3, the other strand is going to be 3 to 5. Okay, so this is the original DNA. And the process of DNA replication basically starts by creating a bubble. Creating a bubble like that. This is the 5, this is the 3, this is the 3, this is the 5. And once you open up the bubble, then you'll be able to see the individual ACTG in there. Okay? And the other side, of course, is complementary to it. And then you can build the missing half. And then the bubble is going to expand sideways. And then you're going to be able to open up the entire DNA. Right? As a loose analogy, is like think of your zipper. Okay. When the zipper is like zipped up, when it's zipped, don't draw this. Okay. If it's zipped up, it's like that. But once you pull on the thing to unzip it, right? Something like that, huh? Okay, when you unzip it like that, then you will have access to the little, I don't know what you call those things, but the little teeth looking thing. Right? You can't see them when it's all zipped up. But once you unzip it, then you can see the individual teeth, right? Individual tooth, that tooth. Sorry. So the base here, it's like the zipper. And then what happens is afterwards, you will basically have the original 5 to 3 here, and you will be building the missing half. This is, of course, 3 to 5. It's always anti-parallel. And then you will have the other half here. Like that. Okay? That's the overview. Now, this bubble here that we create, this bubble here, is called the origin of replication. And it's also called the replication bubble. This picture is just telling you that the eukaryotic DNA is linear, and then prokaryotic DNA is circular. Okay. I guess you don't really need this here if it's in the way. Okay. So after we form the replication bubble, it will expand, and eventually we will be able to create the two separate strands. So what we're going to do now is we are going to look at the replication bubble in more detail. Okay, so I'm going to put the 5 and the 3 here, 3 and the 5. Whole thing is the replication bubble, origin of replication. But this here, one side of the bubble, that's called the replication fork. Sorry for all these terms. Okay, so there is an other fork here, also replication fork. Okay, so I'm gonna zoom in here. Okay, draw this part big, okay? We have a lot of things we need to fit on this. And then we're gonna have the other side here. All right, so I'm gonna stop here for a second, just let you catch up with all the stuff, and let me know if some of the drawing is not making sense. I can explain. All right, so here we created the bubble, and we're focusing on one side of the bubble, which is called the replication fork. And I am gonna actually put some nucleotides here, okay? Um, just try to copy what I have. I mean, I'm totally randomly making it up as well, but it's probably easier for you to just copy what I have than to create your own. And then on the other side, it would have to be complementary, right? C goes with G, G goes with C, and so on and so forth. Okay, so take a moment and fill in the nucleotides. And then over here, we're just gonna put like it's like the unzipped part of it, right? So I'm not gonna put any ACTGs. You can't really see it, right? Like I said, you can do a few more if you want. Doesn't really matter. Okay, so remember the top strand is 5 to 3 in our case, and then the other one is 3 to 5. I'm just gonna be waiting here for a bit for you to catch up. All right. So there is an enzyme here. This is the first enzyme we're gonna learn today. This is like the thing that you unzip your zipper with, the thing that you hang on to. Again, I don't know what you call that, but it will basically move along the DNA and expand the replication bubble. Okay? The name of this enzyme is helicase. Helicase, which unzips the DNA to expand the replication bubble.
It's helicase or helicase?
Uh, Sorry, I'm missing a CMI. Thank you. Helicase. Good eye. Okay, so you would have a helicase right here, unzipping, and then you would have another helicase here, unzipping. So we expand the bubble sideways. So that's pretty self-explanatory, I hope. Now, this next part may be a little bit challenging to draw, so just watch first. Don't erase the 5 and the 3, keep them where they are. Okay, and then this like actually continues, right? And form the double helix. I just didn't draw the double helix because like over here, because oh my goodness, what happened? I don't know what happened. Because if I draw it here, it would be very like confusing, I suppose, very messy. Okay. But then you gotta understand the rest of the DNA is still the double helix. Okay. And here's the thing. When you are trying to unzip something that is already in a coil, when you try to unzip something that's already twisted up, you are going to introduce tension and stress into the system. Yeah, you're going to basically cause it to go into a knot, or like it's going to wound up even tighter over here. And we don't want that because it might break the DNA. So there has to be another enzyme over here, and this is called gyrase. If you read this online, they might sometimes give you another name, like topoisomerase or something like that, but just stick with the easy name here, okay? So gyrase is going to reduces tension that builds up in the DNA as it unzips. Okay? So there you go, helicase, gyrase. 2 down, 6 to go. Next. Now, don't forget, the two strands of the DNA are naturally attracted to each other, right? The C's wants to bind with the G's, and the A's wants to bind with the T's because of the hydrogen bonds attraction. So we want to keep the two strands apart. We don't want them to, close up, and that way we won't have access to the new nucleotides, right? So you want to keep them apart, and that's what these little orange balls are. They are called single-strand binding proteins, and their job is to keep the DNA strands apart. Okay, so these are single-strand binding proteins. Okay, and their job is to prevent DNA from coming back together. Okay, that's all. Any questions so far? All right, last thing, and then we're gonna let you go. We're gonna do it on the side here. We have the DNA polymerase 3. Okay, I know if you look at the list, there's something before this thing. Um, I'll go back to that later, okay? But this is DNA polymerase 3. 3, Roman numeral 3. Okay, so this is the enzyme that builds the new DNA strand. Remember, this is semi-conservative model. So you separate the two pink, so to speak, and then you're going to put new blue ones on, right? Something has to build that blue strand. That something would be the DNA polymerase 3. Okay. Uses the template strand, uses the parental DNA as a template to build the complementary strand. But this guy, this DNA polymerase 3, has two limitations. Two limitations. Okay. Number one, can only build on existing DNA strand. What that means is, just watch for a second, okay? Like here, if it comes here and it doesn't see any nucleotides there, I mean, like you see the black nucleotides, right? But that's like the original strand, right? And its job is basically to use the black nucleotides and build like T, T, A, G, C like that, right? Okay. But it won't do that unless there is already some work done. Okay. So something has to start this. It's not gonna do it on its own. It needs someone to build a strand a little bit first, and then it will continue to work. Does that make sense? Okay, so that's the first limitation. Can only build on existing DNA strand. Something else has to start the process, and then it will continue. It's like my kids, right? They never start their own homework, right? Yeah, I always have to sit down with them. I do a few questions with them, and then they will carry on their own. Right? So this is like that. Something has to start the process first, and then it's going to carry on. Number two, the second limitation, can only build in the 5' to 3' direction. Okay, so it can only go in one way. Next class, Thursday, we will continue this note. But before then, you redraw it if you need to. And you definitely need to understand all this stuff that we have so far before you come to Thursday's class. Okay? We're gonna talk about how these limitations are going to be solved and the consequences of some of these limitations, actually, for humans specifically, because of this particular limitations. Believe it or not, we actually lose a bit of DNA every time we copy it, and that leads to a bunch of other problems. So that's it for today. Thank you. Enjoy the rest of your day, and I'll see you all on Thursday. Bye for now.
Thank you.
Yeah, no problem.